Hormones. The definition of a hormone has been expanded over the last several decades. Hormones secreted by glands such as adrenals, ovaries, parathyroids, pituitary, testes, and thyroids were originally considered to represent all the physiologically relevant hormones. Today, the term hormone refers to any substance in an organism that carries a signal to generate some sort of alteration at the cellular level. Thus, endocrine hormones represent a class of hormones that arise in one tissue and travel a considerable distance through the circulation to reach a target cell expressing cognate receptors. Paracrine hormones arise from a cell and travel a relatively small distance to interact with their cognate receptor on other neighboring cell. Autocrine hormones are produced by the same cell that functions as the target for that hormone. Neighboring cells may also be targets. Thus, we can classify hormones based on their of action. Often, endocrine hormones that travel long distances to their target cells may be more stable than autocrine hormones that exert their effects over very short distances. A diverse array of hormones, each with distinctive mechanism of action and properties of biosynthesis, storage, secretion, transport, and metabolism, has evolved to provide homeostatic responses. Hormone secretion. One, endocrine hormones are secreted directly into the bloodstream. Second, exocrine hormones are secreted directly into a duct. Cascade system of hormone. For many hormonal systems in higher animals, the signal pathway originates with the brain and culminates with the ultimate target cell. The signal may be transmitted as an electrical pulse, action potential, or as a chemical signal or both. In many cases, but not all, such signal forwarded to the limbic system and subsequently to the hypothalamus, the pituitary other gland that secretes the final hormone. Classification of hormone. Hormones can be classified according to chemical structure, mechanism of hormone action. Classification based on chemical structure. Hormones are usually classified into three main groups on the basis of their chemical structures as follows: peptide or protein hormones, amine hormones or amino acid derivatives, steroid hormones. 1. Peptide or protein hormones. Most hormones fall into this class. These are water soluble and may have 3 to over 200 amino acid residues. Example: hormones of the hypothalamus and pituitary, as well as insulin and glucagon of the pancreas. These hormones are synthesized on ribosomes in the form of longer precursor proteins, prohormones, then packaged into secretory vessels and proteolytically cleaved to form the active peptides. Insulin is a small protein with two polypeptide chains A and B, joined by two disulfide bonds. It is synthesized in the pancreas as an inactive single chain precursor, preproinsulin with an amino terminal signal sequence that directs its passage into secretory vessels. Proteolytic removal of the signal sequence and formation of three disulfide bonds produces proinsulin, which is stored in secretory granules, membrane vessels filled with protein synthesized in the endoplasmic reticulum. In pancreatic cells, when blood glucose is elevated sufficiently to trigger insulin secretion, proinsulin is converted to active insulin by specific proteases which cleave two peptide bonds to form the mature insulin molecule and C-peptide, which are released into the blood by exocytosis as shown in figure. Second, amino acid derivatives. These are small water-soluble compounds containing amino groups, for example, adrenaline of the adrenal medulla, thyroid hormones. catecholamine hormones. The water-soluble compounds epinephrine, adrenaline, and norepinephrine, noradrenaline are catecholamines named for the structurally related compound catechol. They are synthesized from tyrosine. Catecholamines produced in the brain and in other neural tissues function as neurotransmitters, but epinephrine and norepinephrine are also hormones, synthesized and secreted by the adrenal glands. Like the peptide hormones, catecholamines are highly concentrated in secretory vessels and released by exocytosis, and they act through surface receptors to generate intracellular second messengers. They mediate a wide variety of physiological responses to acute stress. The thyroid hormones T4 thyroxine and T3 triiodothyronine are synthesized from the precursor protein thyroglobulin. Up to 20 Ti residues in thyroglobulin are enzymatically iodinated in the thyroid gland. Then 2-iodotyrosine residues condense to form the precursor to thyroxine. When needed, thyroxine is released by proteolysis, condensation of monoiodotyrosine with diiodothyronine products T3, which is also an active hormone released by proteolysis. Thyroglobulin tire goes to thyroglobulin tire, iodinated tire residue. On proteolysis gives thyroxine T4 triiodothyronine T3. Steroid hormones. These are fat-soluble, lipophilic, and all are derivatives of cholesterol. For example, adrenal cortical hormones, androgen, male sex hormones, estrogens, female sex hormones. Steroid hormones. The steroid hormones, adrenocortical hormones, and sex hormones are synthesized from cholesterol in several endocrine tissues. Cholesterol into progesterone and it converted into cortisol, glucocorticoid, aldosterone, mineral corticoid, and testosterone. And testosterone goes to estradiol sex hormone. They travel to their target cells through the bloodstream bound to carrier proteins. More than 50 corticosteroid hormones are produced in the adrenal cortex by reactions that remove the side chain from the D ring of cholesterol and introduce oxygen to form keto and hydroxyl groups. Many of these reactions involve cytochrome P450 enzymes. The corticosteroids are of two general types, defined by their actions. Glucocorticoids, such as cortisol, primarily affects the metabolism of carbohydrates. Mineralocorticoids, such as aldosterone, regulate the concentration of electrolytes K plus 1 Na plus calcium 2 plus Cl negative in the blood. Androgens such as testosterone and estrogens are synthesized in the testes and ovaries. They affect sexual development, sexual behavior, and a variety of other reproductive and non-productive functions. Their synthesis also involves cytochrome P450 enzymes that cleave the side chain of cholesterol and introduce oxygen atoms. All steroid hormones act through nuclear receptors to change the level of expression of specific genes. They can also have more rapid effects mediated by receptors in the plasma membrane. Classification based on mechanism of hormone action. Hormone can be classified according to mechanism of hormone actions to group 1 hormones and group 2 hormones. This classification is based on location of the hormone receptors. First, the hormones of group 1 are lipophilic, which readily pass through the lipophilic plasma membrane of the target cells and interacts with receptors which are located intracellular in either the systole, or the nucleus. The receptors for the different steroid hormones are found mainly in the cytoplasm, and the receptors for the thyroid hormones are found in the nucleus. Second, the hormones of group 2 are water soluble hormones which do not penetrate lipophilic cell membrane readily. The receptors for such hormones are located on the outer surface of the target cell surface receptors. Mechanism of hormone action. The first step of hormone action is binding of hormone to specific receptor of the target cell. Hormone receptors complex activates the hormonal effects. Hormonal receptors are large proteins which are highly specific for a single hormone. Due to this specificity, a particular hormone will act on a particular tissue. Mechanism of hormone action at systolic nuclear level. Several hormones, example steroid hormones, adrenal and gonadal and thyroid hormones, bind with receptors inside the cell rather than in the cell membrane. Because these hormones are lipid soluble, they rapidly cross the cell membrane and interact with receptors in the cytoplasm or nucleus. The receptors for the steroid hormones are found mainly in the cytoplasm, and the receptors for the thyroid hormones are found in the nucleus. Cell membrane receptor of hormone action. The receptors for group 2 hormones are located on the outer surface of the target cell because these hormones are water soluble which do not enter lipophilic cell membrane. Hormones that bind to surface receptors of the cells communicate their action through intermediary molecules called second messenger. The hormone itself is the first messenger. On binding of the hormone to the receptor, a conformational change occurs in the receptor that causes activation of the G protein that consists of three subunits alpha, beta, gamma. GDP, inactive G protein, convert to GTP, active G protein, interacts with the other intracellular signalizing enzymes such as adenylate cyclase which generates CAMP for many hormones, example epinephrine, glucosan, calcitonin, PTH, etc. Phosphatidylinositol, calcium second messenger. Certain hormone receptor interaction result in the activation of the enzyme phospholipase C through a specific G protein. Phospholipase C enzyme catalyzes the breakdown of phospholipids in cell membrane, especially phosphatidylinositol biophosphate PIP2 into inositol triphosphate IP3 that liberates stored intracellular calcium ions from mitochondria and endoplasmic reticulum. The calcium in turn acts as a third messenger which influences a variety of biochemical processes. Diacylglycerol DAG. The other second messenger activates the enzyme kinase C which then alter physiological processes. The hormone thyrotropin releasing hormones TRH, gastrin, choleocytokinin act through the second messenger.